All right. Well, thank you, everybody. Today, I want to keep this to be as basic and straightforward as possible because it really is now is the time that we're really starting to see some of these strange things show up in our field, and we're starting to get a lot of questions about what could be the potential causes of some of the things that people are observing. And one thing that we have to keep in mind is that there are many different diseases and other things that are not diseases that can cause some of these symptoms and impact the crown, stems, and foliage of our soybeans in Illinois. And because of how they affect the plant and what they're doing, a lot of them have shared symptomology. And so what I want to do today is kind of go through the process of what you should do when you're seeing something strange in your field and the sorts of things you should look for to try to give yourself a better idea of what the potential cause of the problem could be. Because one of the biggest problems you can have as an agronomist or a CCA or a grower is misdiagnosing a disease and saying it's one thing when it ends up being something else, because it can result in a lot of unneeded inputs, incorrect cultivar variety selection, all sorts of different things. And so I give the example, perhaps you think you have sudden death syndrome, but really it was just fungicide toxicity. So you're investing the next time you go in there to manage sudden death syndrome when really that's not a problem. It was just simply the product and the time of the year when the product was applied that was the issue. So these are some of the reasons why we want to go through this today. And this is kind of the sort of thing that we're seeing right now, right? We're driving by our fields, we're noticing, okay, well, there's some yellow patches showing up there. That looks kind of funky. And so maybe we go out in the field and we see things like this. So maybe the leaves are turning yellow, maybe the leaves are defoliating or plants are dying, or perhaps you get this symptom here with the interveinal necrosis and chlorosis. And one of the things that I really want to kind of nail home today is if you see something like this in your field and you go out and you start seeing foliar symptoms, that is not what you should be using to diagnose the issue or the problem. And as you're going to see here, many of these diseases, many of these issues can cause very similar effects on the foliage. So just because you see green veins with yellow or brown tissue in between does not indicate necessarily that you have sudden death syndrome. I think that's the one thing a lot of people like to attribute that symptom to. So what could some of these problems be? Well, it might be that it was, it could be soil properties. Maybe it's compacted areas or sandy areas. And so those plants are stressed out and you're getting some drought. You could have nematodes. You could have several different diseases here, as we're going to talk about in a second. Also things such as fungicide toxicity and also natural senescence oftentimes can result in some of those patches. You might come by one day and think, boy, that's strange. What kind of issue do I have? And then maybe you come back in another week, your whole field starts to senesce, and it's just that those areas perhaps had a different soil type. They were a little drier, and so those plants start to maybe senesce a little earlier, and that's why it was showing up. So this is the time of year when we start to see all these different things blending together, and that's why it's important to know how to figure things out. Another thing that's important to keep in mind, especially when it comes to diseases, is a lot of times you're going to see more than one of these occur in a field at a given time. So it is important to go out into the field and actually do a good job of sampling. And one of the things I want to talk about here is kind of what's the process we should take. So as we're driving by our field, first you want to make sure that you're looking at that pattern in the field. Is the entire field affected? Is it strips? Is it in blotches, kind of patches? Note those patterns. And then you're going to actually want to have to get out of your truck. And I know a lot of times this is difficult to do. We have lots of acres to look at. Fields are very large, but if you see something that looks out of whack, pull off, take the time to get out of your truck and actually look at some of those plants, bring them back into your truck or into your office and start looking for some of the things we're going to discuss. The foliar symptoms in the infected areas are going to be important, so keep track of the types of symptoms do you see. Are you looking at yellow leaves on the plants that are somewhat uniform, interveinal necrosis, dead plants, all those sorts of things. And then you're going to want to get out and dig up those plants and take as many samples of those symptomatic plants as you can, but also try to grab some healthy ones so you have something to compare to. And the reason you need to take many samples is because oftentimes some of these signs and symptoms that I'm describing for these diseases are not going to be observable in every plant. So you do need to have several plants observed before you might actually see those signs and symptoms. And that just has to do with the fact that biotic, living organisms grow at different rates. They infect plants at different rates. And so a lot of those symptoms and signs of the pathogen may not be observable all at the exact same time. So you do want to grab several plants to look at. And then what you're going to want to do is you want to look at the roots and the stem and crown with the hand lens that you hopefully all own. And that's very important because in order to see a lot of the structures that I'm going to show in these pictures, unless you've had LASIK surgery, you have very good eyesight, a lot of these are going to require the assistance of a 10x, 20x hand lens to really help you visualize those structures. And then the last thing that you're going to want to do, and this one's actually very important when you're trying to separate out the cause, the actual cause, is splitting the stems. And so splitting the stems is something that I say is probably the most important step with a lot of these, and you want to make sure you do it correctly. The way I do it is I tend to focus on the lower 5 inches or so of the stem through that crown. And what I'll do is I'll whittle away with my pocket knife and try to split the stem. Things you should always have in your vehicle when you're going out there, just so you're prepared and able to properly scout your fields. You want to make sure you have some sort of shovel or a spade. Doesn't have to be anything crazy, but the key here is you probably, unless it's a very sandy soil or it's very moist, you're probably going to want to dig those plants up so that you can shake off the soil. A lot of those structures and a lot of those signs and symptoms are going to be on the roots and that lower crown. And if we just rip up the plants, we're going to lose a lot of that information that might help us make a proper diagnosis. A knife, I like to say, don't get crazy with your knives. I like just a little pocket knife, but I know, people use different things. The idea here, you're going to need that to split the stems and also cut those plants at the lower base. And then this is a type of loop or hand lens that you might want to have. So hopefully everybody has one of these. These are often handed out at meetings and other get-togethers. So keeping one in that center console or in your glove compartment can really help you here with making some diagnoses here. I also say really important things to keep as well, gallon Ziploc bags and permanent markers. If you find something interesting or you aren't sure of what you have, you're going to want to send these off to, say, a diagnostic clinic like the University of Illinois Diagnostic Clinic or Iowa State or wherever you may be. And so having Ziploc bags on hand and markers where you can write that down can be very helpful, especially if you're collecting many samples over the day. So those are little things you should have in your vehicle just so that you can be prepared to properly sample your fields. Now, when you're looking out of the truck, remember I told you the first thing you should look at is the pattern from the road. What is that pattern? Is it the entire field? Is it strips or streaks, boxes, squares? All these different things tend to indicate that this is an abiotic issue. So this is not a disease. This is due to some sort of, some other issue, okay? And those are some examples there: nutrients, soil type, natural senescence, fungicide toxicity. All these different things are going to be caused by, it could be a sprayer application, it could be the variety, but the one thing we have to keep in mind is biological organisms, living organisms, aren't going to show symptoms in these clearly structured and defined shapes like squares and strips. They're typically going to be associated with low-lying, high-lying parts of the field, little patches, little blotches. Those sorts of shapes are going to be more indicative of a biological disease. Also, diseases caused by biological organisms do not show symptoms all at once. Typically, it tends to be a gradual plant-by-plant progression over time, versus these abiotic issues, a lot of them are going to be showing those symptoms more uniformly. Now, the one thing I was talking about, the importance of not just looking at the foliage and making a conclusion or coming to the conclusion that you're dealing with sudden death syndrome. is fungicide toxicity. Now, this might not be something that we see in a lot of parts of the state, but maybe further down in the southern parts of the state, we might see some of this. Typically, this is going to happen when we're spraying specific products on our soybeans, on that foliage, on really hot days. And then if it's really hot or dry and windy after that application, what happens is that fungicide gets taken up into the tissue really quickly, and it actually causes a burn on the foliage and interveinal necrosis, which can look a lot like sudden death syndrome and some of these other diseases we have. A lot of these symptoms also, in addition to being associated with the product, can be variety dependent. So some varieties are more susceptible than others. And because this is associated with a fungicide application, usually those symptoms are going to be shown up fairly uniformly across the field, across the canopy within 1 to 2 weeks after the spray, sometimes sooner, depending on the environment. But keep in mind, this is going to be uniform in distribution, and it's going to be uniform on those plants. Now, these are some examples of chemistries or fungicide products that are known to cause phytotoxicity in soybeans. This is an older list. The thing to take home about these is that these products all contain at least one triazole, and it's that triazole, by and large, that is causing that symptom on those leaves. And so the big three here that can cause problems: tetraconazole, prothioconazole, flutriafol, and also tebuconazole. Those tend to be the ones that can cause that burning type symptom. So some of our newer products are going to have some of these as well, and potentially could cause some of those symptoms that you might see in your foliage. So if you do see symptoms develop uniformly across the field, go look at when the spray was made, and look at the product and see what the active ingredients are that could at least point you in the direction of potentially having phytotoxicity in your soybeans. Now, what are some biological issues that cause interveinal necrosis and chlorosis? Okay, so these are the diseases that we can encounter or have been encountering here in 2020. Now, the one that everyone's probably most familiar with is sudden death syndrome. This is a fungal disease caused by Fusarium virguliforme. It's favored by wet conditions early in the season, usually right around planting. And then later in the season, usually after R3, if we've got dry conditions and maybe fluctuating rain, what that does is it results in the fungus that had colonized the roots of those plants earlier to produce a toxin that moves into the foliage, and it accumulates in the foliage. And that's why you start to see that chlorosis or yellowing and browning of the tissue in between those veins, which will remain green. When you cut your lower stems, okay, so that lower part you're whittling away with your giant knife that I showed you earlier, that lower stem and taproot will have a brownish discoloration on the outside of the pith, but the inside of the pith will remain white and healthy. That's very important. The other thing is this will cause a rotting of the roots, so the roots will not have that nice white appearance. They'll be kind of a brownish, even black sometimes, depending on the severity. You might also see a bluish growth on the lower crown or roots. And if you do see that bluish growth, that's pretty much the, I like to say that's the home run sign of sudden death syndrome. So I have, here is, and this isn't showing up very good, but this was kind of a light blue color on this close-up of a root that I was looking at in a field here. And again, this bluish color here on the root. What that is the fungus and the spores of that fungus being produced on the roots. Sometimes you'll see it on the lower stem as well. One thing to keep in mind too, when these leaves do start to defoliate, the petioles here, the center bit of xylem on those leaves will stay attached to the plant, but the leaves themselves will defoliate and fall off. And you can kind of see what that looks like here. They look kind of tattered and withered, okay? Over time, that's what they're going to do. So that's sudden death syndrome. The next one that I want to talk about is red crown rot. Now this is a newer one that we're starting to look at in Illinois. I'll talk a little bit more about this here in the upcoming slides in terms of where we're seeing it. But this is kind of the redheaded stepchild of SDS. Very similar conditions favor red crown rot. It's caused by a different soil-borne fungus. Likes it wet early and again dry or wet after R3. we start to see a lot of the same symptoms being produced. So interveinal necrosis, those plants will start to blight and die, just like sudden death syndrome. But unlike sudden death syndrome earlier, where I showed you these petioles staying attached, these leaves will remain attached, as you can kind of see in this image I'm showing here, those plants, and they die. They just kind of turn brown and those leaves hang on to the plant. The other thing is, later on, especially if you find plants that are pretty far along in terms of their senescence, and after some wet weather, you dig up those plants, shake off the soil, and look at the base of the stem, and what you're going to see are these red pinhead structures on the lower crowns and roots. Now keep in mind, this is where a hand lens is very helpful, because a lot of times, depending on the variety and what's going on, basically your stems can have kind of a reddish look to them, but you're not going to necessarily see these structures. And just because you see what looks like a reddish color doesn't necessarily mean you have red crown rot. But if you see these little pinhead structures along with these other symptoms I'm showing you, that's more indicative of red crown rot. Also, when it's very wet, you'll see a lot of mycelium or white mycelial growth on the base of these stems. Similar to SDS, your roots are going to be rotted as well. They can have a black appearance to them. And this is what you might see if you're able to zoom in a little more. So this, when I say these little pinhead structures, this is what I'm looking at here. These little red structures along the root. They're pretty tiny. Oftentimes, like I said, they can be at the, underneath the soil, but they can also be at the base of the stem. Another important thing, like I mentioned before, multiple things can be found at once. In this case, we have SDS and red crown rot on this particular plant here, right on the base of the stem. So that's what you want to be looking for with red crown rot. Use that hand lens. Right now, we're kind of looking at the distribution. We found fields, several fields within Pike, Henderson, and Madison counties that we know have red crown rot just based off of the signs and symptoms. We have other ones that are suspect, but we need to utilize a different type of tool to actually confirm that that fungus is in those tissues. It's very difficult to isolate this out if the structures aren't present on the base of the stems. We've also had several false alarms. And for example, we had a grower who thought he had red crown rot. Last week I went on into his field, did some sampling, started whittling away at the stems, and he actually had brown stem rot, which is actually what he thought he had, but he thought, boy, those stems look red, maybe it's red crown rot, and sure enough, it was brown stem rot. So we've had a little mix of everything. But right now, where this circle is, that's kind of the area where I'd say we're most likely to see it right now, but I don't know how widespread it is. And at the end of this presentation, I'll have some information if you do see interveinal necrosis or things that you think could be red crown rot. We're doing a survey as part of our Iowa Soybean Board funded project, and I'll have some information on that at the end of this discussion. Now, I mentioned brown stem rot in the last slide, and that is one that we've seen pop up in a few fields this year in the northern part of the state, as well as here in the central part of the state lately. Now, brown stem rot is caused by a fungus that doesn't overwinter particularly well in the absence of soybean residue, but it likes it wet. And we tend to think of this as a cool season disease. So when it's cool and wet, all the way through that early vegetative phase, that's when we tend to see the most significant infections by brown stem rot. There are two different types of this pathogen. There's a type A and a type B. Type A is the aggressive one, and that's the one that's going to produce those foliar symptoms that you see right here with the interveinal necrosis and chlorosis. Again, looks very much like red crown rot or sudden death syndrome, okay? Type B is one that's less aggressive, and typically we'll only see that in the stems, although those plants, they might look a little off, they might be stunted, and they might develop some poor yields in certain areas. But those are stems only. So we do tend to have aggressiveness, resistance to the aggressive form, and a lot of the PI88788 soybean cyst nematode resistant soybeans that we have. In type B, there's not really as much resistance. And there's also some other particular varieties like Peking and Hartwig sources of SCN resistance that tend to be more susceptible to brown stem rot. So if you see that you have a particular cultivar that's susceptible, you might want to think about switching into a different cultivar next year and look at the source of resistance as well as rotating. And I'll have some information on management later, but really today I want to focus on diagnosing these or identifying these in the field so that you can make management decisions the next year. That's really what's important. Now, when you're going out and you're looking at these plants, again, interveinal necrosis, pull those plants out, split those stems, and here's an example of the stem I split. What you can see with brown stem rot is that lower stem will have a chocolate brown appearance that often has what I call laddering. See how that kind of looks like a ladder you're climbing up? That's indicative of brown stem rot. Sometimes you can have insects such as Dectes stem borer that can cause this sort of symptom as well. Dectes stem borer, oftentimes you'll come up and you'll actually find the pupa hanging out in there somewhere, as well as frass, and it looks a little bit more like sawdust, and sometimes some exit holes as well. Oftentimes not as widespread as you might see here with our brown stem rot. The other key piece to the puzzle here is those leaves again. So they have this interveinal necrosis, but again, those leaves kind of hang on to the stem. So in this way, very similar to red crown rot, but again, it's that inner pith, that inner stem is separating it from red crown rot, where it has those kind of red balls on the outside of the stem and a root rot, okay? Now, now stem canker is one that is a little more complex than I'm going to make it out to be here. It's caused by several different species of Diaportha. It's another fungus. This one really likes it wet, especially during the early vegetative stages. So you can kind of see a common pattern here. If we have a lot of wet weather early on in our soybean growing season, at the end of the season, we're more likely to encounter a lot of these stem and crown issues that I'm describing. Now, historically, we've defined these as being either northern or southern stem canker. I think some of the work that's being done is showing that it's more complex than that. But the key thing is, when we are looking at trying to at least categorize whether or not it's likely to be northern or southern, there are some symptoms that differ between the two. That can kind of help us decide what we're looking at. So the first thing that you're going to see is that with southern stem canker, so typically warmer climates, but we are seeing this kind of moving a little further north into regions, into the state. Southern stem canker will produce interveinal necrosis. This is an example of what it can look like. Some people say that this sort of starts on one side of the leaf and then develops across over time. I haven't observed that very frequently, so I don't know if that's a very good diagnostic character. But the difference that you're going to see with southern stem canker and northern stem canker is that this is a disease of the stem that starts at the nodes. So southern stem canker typically is going to be found in the lower portions of the stem, and you're going to notice these lesions that will kind of form unilaterally up one side of the stem. And they'll develop, or they'll become a little sunken over time. With northern stem canker, typically what you see is the development of lesions right around those nodes, and they'll start as these little spots with, right around those nodes with little black margins, and then they'll expand up and down over time, kind of resulting in this sort of symptom that you see here on the stem. Now, you won't see this sort of interveinal necrosis with northern stem canker, but with southern stem canker, again, you can see it. You're not going to see any kind of root rots in this case. You're not going to see any structures on the base of the stem or on that crown or on the roots. You're focusing right here on the outside of the stem. That's what you're going to be looking for. Now, there are some other things that can cause issues. Maybe they're not going to cause that interveinal chlorosis. So I already mentioned northern stem canker, but I didn't want to separate that one out. But there are some other things that can cause just a general chlorosis of the plants in patches. And sometimes you might see that these are going to be uneven. So one plant will have it, maybe a couple won't. It'll develop over time. It's associated with particular parts of the field, lower high ground, heavier dry soils, okay? So this is something that's not senescence, because the other parts of the field, maybe they remain healthy, but the parts that are affected start to go down more and more over the course of a week or two. The first one I want to talk about are nematodes, and nematodes are a bit frustrating. I know you've heard a lot about them in soybeans, but the two nematodes that are the easiest to diagnose in your fields are going to be soybean cyst nematode and root knot nematode, because they produce structures on the roots. What you do see in severely affected varieties or very susceptible varieties is, and again, this is from my colleague Travis Foskey, he's looking at southern root knot nematode variety trials here, and you can see where those susceptible varieties are. But you can imagine a field, you have a low-lying area or a very sandy area being affected by nematodes. Those plants might be stunted, they might become chlorotic, and kind of develop in these kind of, usually it's an oval patch or an irregular type patch. They follow where those nematodes are. You also can have, unfortunately, other nematodes that don't produce structures on the roots. And those, if they're at high enough numbers, can produce other symptoms as well. And really the only way to get a good idea of if you're dealing with nematode issues is to go out there and sample affected areas versus unaffected areas and mail those off or send those off to a plant diagnostic clinic like the University of Illinois Clinic, or a nematode service that there's several independent ones. You know, go to whichever one you're used to going to and get some numbers and see if there's differences in those nematodes in those affected versus unaffected fields. But nematodes, they usually like the drier areas of the field, lighter soils. As I mentioned, the plants are stunted. If you get out that hand lens again, you dig up those plants, shake off several plants, you're going to notice these little white structures, and they can also have a kind of like a lemon shape to them. They can turn color to be almost a gold or a brown over time as they mature. Those are cysts of soybean cyst nematode. And with root knot nematode, it causes these galling type symptoms here. And so those nematodes go in and they cause those roots to overgrow and gall up. And they're just going to be very erratic and almost carrot-like in their structure. So we did have a couple of fields this year with some severe root knot nematode issues, very impressive galling. Because of the damage it's causing to the roots, when you end up with those droughty type hot weather, you know, these plants aren't able to pull up nutrients and water as well. And so that's why you start to see those symptoms develop, the chlorosis, leaf defoliation, those sorts of things. Here's a link to the plant clinic if you ever do have some interest in sending nematode samples, if you think you do have nematode issues, and they can process those for you. Another one, this is one where we've actually had a couple of calls today, charcoal rot. This one is caused by another soil-borne fungus with a pretty wide host range, Macrophyllina phaseolina. This one likes it really hot and dry when it's during those reproductive phases. Again, this year we had it wet early in a lot of areas, and then we also had some very pronounced droughty, hot conditions in some areas. And so what you'll see is these plants will develop a chlorosis, but then they can blight and die in patches very quickly. So this is an example of what that might look like. And we had some images that were sent to me this morning that I'm pretty sure of charcoal rot. If you get out there and you start looking at those plants, what you'll see is very tiny pin-like structures, oftentimes on the outside of the stems. And if you split those stems open, what you're going to notice, and again, here's what I want to show you. You're going to notice, this is magnified, those little peppercorns are going to be in the center of those stems as well. So remember that laddering we saw in brown stem rot? Inside here, you're going to see a gray fuzziness, and when you zoom in on that with your hand lens, you'll see these little black structures. That's overwintering structures of the Macrophyllina fungus, and that's going to be indicative of charcoal rot. This is what we call a hot season wilt. One thing to keep in mind, in the past, we used to say that these little zone lines that we're seeing here on these stems were due to charcoal rot. These are actually another pathogen called, well, actually it's Phomopsis or Diaportha that cause these sorts of zone lines here. So these are structures of the fungus in here, not charcoal rot. So I'm not going to talk too much about that. That's more of a pod and stem blight type thing, which I'm not going to talk about, but just keep in mind, if you see this sort of thing in your stems, It's not uncommon, but it's not due to charcoal rot. So here's kind of what I tried to whip together if I was going out in the field, the sorts of processes that you might go through in your head to figure out what you might be dealing with this year, because now's the time when we're seeing a lot of these issues. So driving by in my truck, I see something weird, and the first thing I'm going to look at is that pattern. Is it defined pattern, or is it sporadic or not defined? And that's going to tell us, is it potentially an abiotic issue or is it a biotic issue? If I go out there and I look at my plants and there's interveinal necrosis, that might indicate some sort of fungicide phytotoxicity. And again, that's something that tends to show up a little earlier in the season after doing a spray. If you see general chlorosis, that might just be senescence, a soil issue, and drought. And you just want to kind of sit back and wait another couple days and observe. If they're sporadic or not defined, that's going to indicate that you have a biotic disease. So if you have no symptoms or signs on the stem, you split that stem and the lower pith is white, or that lower pith is brown. If it's white, that's SDS. Other things to look for: blue growth, chlorosis, interveinal chlorosis, and the retention of those petioles. If those leaves are retained, you have interveinal chlorosis, you're dealing with brown stem rot. Another thing, yellow foliage, but you may or may not have knots or cysts, so you may have a white inner pith, but you might see some other things on the roots or nothing that would indicate it's abiotic. That means you might have to deal with a nematode issue, and then you should be sending those off to have that confirmed. The last thing here, symptoms or signs on stems. If you're looking at the outside of your stems, you see those little black peppercorns on the stem, and cut those stems within it, you see those. Charcoal rot, again, very fast blight in that particular disease. If you're seeing small red peppercorns on the lower stem, as well as on those roots, then you're probably dealing with red crown rot. You're also going to see interveinal chlorosis, and the leaves are going to be retained. And then if you see dark blotches at the nodes, maybe you see unilateral growth on the outside of the stem, and those lesions are become sunken over time, that's going to be stem canker. And if you have southern stem canker, you may see some interveinal necrosis. This is what I call my quick management cheat sheet. This is something we can send you after this, but just keep in mind, one of the reasons we need to make accurate diagnoses of our issues is because different diseases have different management strategies that can be effective or ineffective. And so that's why we need to make sure we're actually getting out in the field and looking at everything that we have. And my final thing here is my last slide. Just want to talk a little bit about the survey we're doing for red crown rot, because this is a newer disease that we have in the state, and it does show symptoms very similar to SDS, but can't be managed like we do SDS. We want to look and see how much we have in the state, and basically we're looking for samples of plants that have interveinal chlorosis, and maybe you think they have red crowns, or you actually see the structures of the fungus. It's even better. So basically, if you can go out, we're looking for people to go out and try to grab 4 or 5 symptomatic plants from the field and just send us the lower stems and the roots, place in a plastic bag, and try to ship them up to us within 2 or 3 days, and keep them in a fridge or cool during that time. Make sure to include your name, county, GPS, previous crop, and cultivar on the bag, and then you can mail those to us. And then what we'll do is we'll include those in our survey. And so keep in mind, this is a graduate student project. She's a new graduate student, so this isn't a clinic service. So we will get the data to you hopefully by the winter, but it's not going to be a 1-week, 2-week turnaround, but you'll definitely have some information that we'll share with everybody and individuals here once we're done processing all those samples.